Hi everyone, this is for Chapter 2, Segment 2 in our Behavior and Neuroscience class. This chapter, and especially this segment, I feel has a lot of information, so I'm just going to do my best to kind of pick out the key aspects that are probably most important. This section does start with glial cells. Glial cells are kind of like glue, so to speak. They were originally thought to hold the nervous system together. As we did more research, though, we found out that glial cells do communicate with each other and with other neurons, so they're more so than just glue. Glial cells actually affect neuronal functioning by providing neurons with raw material and chemical signals. All of this alters the neuron structures and excitability. And although there are at least as many glial cells as there are neurons in the brain, glial cells are slightly less complex, only coming in 4 basic forms, only 3 of which are actually found in the brain. The first glial cell found in the brain is an astrocyte. An astrocyte is a star-shaped glial cell, and it has numerous extensions that run in all directions, so that it kind of branches out like a star. Highlighted there, the astrocytes actually have sucker-like feet. These sucker-like feet connect on the blood vessels. This allows the blood vessels the astrocytes to help regulate local blood flow, and it provides more supplies to the neurons when they are active because it gets anything they need from the blood. Astrocytes weave among neurons. They receive synapses directly from neurons, and they surround and monitor the activity of nearby neuronal synapses. They do account for 17% of glial cell, and there is evidence of crosstalk among astrocytes and neighboring neurons. This is what we call a tripartite synapse, and it is where astrocytes directly participate in transmitting information between neurons. This type of gliotransmission, so to speak, however, remains controversial. And it is important to remember that astrocytes help with the formation of new synapses, in addition to pruning surplus synapses, which basically means getting rid of the axis or dead synapses that the brain no longer needs, and it's actually a very normal and useful part of brain development. The next one is microglial cells. They are extremely small glial cells that remove cellular debris from injured or dead cells. So although they are very small, they are actually nicknamed the brain's cleanup glue. They are also known as microglia. Then these microglial cells form a spherical containment zone around the site of debris or injuries, and they are very active cells despite their small size. This image shows just the microglia encircling or engulfing the debris. And they account for 7% of glial cells. We are now realizing, however, that the microglials are more than just the brain's cleanup crew, because they do appear to be a key component in neuronal pain systems. So moving on to the last one, they kind of combined the oligodendrocytes and the Schwann cells. This picture is of an oligodendrocyte, and they are responsible for forming myelin in the central nervous system, so your brain and your spinal cord. And they provide myelin beads to several nearby axons. More than 75% of the brain's glial cells are oligodendrocytes, and oligodendrocytes provide tropic signals that enhance the structural integrity of axons. Schwann cells, on the other hand, form myelin in the peripheral nervous system, or the rest of your body. A single Schwann cell ensheaths a limited length of an axon, so it is smaller compared to the oligodendrocyte, but no matter where the myelination process is coming from, myelin still increases the speed in which electrical signals pass down the axon, so it's very useful. There are actually many short axons that lack in myelin but are still surrounded by oligodendrocytes or Schwann cells. In that instance, the cells segregate the unmyelinated axons. So when talking about the two, it's very important to remember that both do perform myelination, which is wrapping the axon up in myelin. They just do so in different nervous systems. The oligodendrocyte is for the CNS, and then the Schwann cells are for your peripheral nervous system, which we're actually going to talk about in just a second. A couple more notes just to touch on, though. Myelination, as mentioned before, is the process of myelin formation. It is actually a very complex process that continues for a long time in humans. In some brain regions, it lasts for 10 to 15 years after birth, and in others, it occurs throughout the lifetime. Myelin is the fatty insulation around an axon, which is formed by glial cells, and myelin basically just improves the speed of conduction of an action potential. So keep in mind, when talking about glial cells, one role is to insulate and isolate synapses to prevent one from affecting the other. This is best seen in myelination. Glial cells are actually very significant for clinical interest because, unlike neurons, glial cells divide throughout the lifetime. Because of this continuous division, glial cells actually form many types of brain tumors. This is seen when glial cells swell in response to brain injury. It's especially common in astrocytes. This produces a type of edema, which is swelling of tissue such as brain in response to injury. This edema damages neurons, and it's responsible for many symptoms of brain injuries. Astrocytes actually directly influence local brain chemistry, and they factor in diseases that cause change in neuronal excitability. This is commonly seen in epilepsy. Microglia are implicated in degenerative processes or many commonly known symptoms of Alzheimer's. This is because the changes in the microenvironment of the microglia can cause them to dangerously inflame. Multiple sclerosis, on the other hand, occurs from the loss of insulating myelin sheaths in various brain regions. The consequences for the individual depends on the region that is impacted by the missing myelin sheath. Changes in all three kinds of glial cells, however, especially in oligodendrocytes, are implicated in the onset and symptoms of schizophrenia. So, moving on from there, we're going to talk about the nervous system. The nervous system consists of two divisions. One is the central nervous system, which is your brain and your spinal cord, seen in blue on the diagram on the top of page 36, while the peripheral nervous system is everything in yellow, or the rest of your body around it. The peripheral nervous system courses through the body and connects all body organs and systems to the central nervous system, or the CNS. The peripheral nervous system is actually then broken down into two divisions. The first division is the somatic nervous system, which is the part of the peripheral nervous system that provides neural connections to the skeletal musculature, or in very simple terms, it connects our brain and muscles to our head senses. The somatic nervous system is responsible for the sensory systems of the body. It's important to remember. The second division is the autonomic nervous system, or part of the peripheral nervous system that supplies neural connections to glands and to smooth muscles of internal organs. This is mainly called the viscera. Viscera just refers to your internal organs. So, gross neuroanatomy is then used to describe the anatomical features of the nervous system that are apparent to the naked eye. So, that talks about the CNS and peripheral nervous system because we could see all these structures without a microscope. A nerve is then a collection of axons bundled together outside the CNS, so nerves are found in your peripheral nervous system, and they extend throughout the body. A motor nerve conveys neural activity to muscle tissue and causes it to contract. So motor nerves are the nerves that transmit information from spinal cord and the brain to muscles, organs, and glands. A sensory nerve, on the other hand, conveys sensory information from the periphery into the central nervous system. So sensory nerves basically convey information from the body to your brain. The somatic nervous system is then composed of cranial and spinal nerves. The somatic nervous system is the main pathway from which the brain controls movement and receives sensory information from the body/sensory organs of the head. So cranial nerves are connected directly to the brain. There are 12 pairs of cranial nerves. One on the right corresponds to the one on the left, and vice versa. Each nerve is known by name in Roman numeral, and they are formed by bundles of axons. Cranial nerves serve as the sensory motors for the head and neck, and then each nerve passes through small openings in the skull, which allows them to enter and exit the brain without actually ever touching the spinal cord. A spinal nerve, on the other hand, is a nerve that emerges from the spinal cord. They're also called somatic nerve or body nerve. The figure on the top of page 37 highlights the difference between cranial nerves that are sensory pathways, motor pathways, and then cranial nerves that are both sensory and motor pathways. So this is very useful to look at. I'm just going to quickly mention that the 3 cranial nerves that are involved in sensory pathways, so the ones seen in blue, are olfactory, optic, and vestibular ocular. The 5 cranial nerves that are involved in motor pathways are red. They're ocular motor, trochlear, abducens, spinal accessory, and hypoglossal. The 4 cranial nerves that are both sensory and motor pathways, so you see some boxes share both red and blue. They are trigeminal, facial nerves, glossopharyngeal nerves, and vagus nerves. It's important to keep in mind that there are separate axons within the nerve which carry motor or sensory information, so it's not from the same one. Spinal nerves, on the other hand, they come in 31 pairs and they emerge at regularly spaced intervals throughout openings in the backbone, which the figure on the top, page 38, does a really good job of highlighting. One member of each pair serves each side of the body. So each spinal nerve consists of the fusion of two distinct branches, or roots, which are functionally different. The dorsal root is the branch of the spinal nerve that carries sensory information from the peripheral nervous system to the spinal cord that is found in the back, which can be seen right in there. And then there's the ventral root, which is the branch of the spinal nerve that carries motor messages from the spinal cord to the peripheral nervous system, and that is located in the front. So the ventral root is basically just responsible for projections of the spine to the muscles. This figure also shows that the spinal column runs from the base of the brain into the tailbone, and that the spinal cord is surrounded by bony vertebrae that is enclosed in 3 membranes. So fibers from different spinal nerves join to form peripheral nerves, and each spinal nerve is named according to the segment of the spinal cord that is connected to. So as you look over here, you see that we have the cervical, the thoracic, the lumbar, the sacral, and the coccygeal. And then the naming that we refer to these segments affects the position of the spinal cord that they're in. So if we take the thoracic one, for example, if we are referring to the 12th segment of the thoracic portion of the spinal cord, we call this T12. T for thoracic, 12th segment, so on and so on. We do this with all of them. So getting back to your autonomic nervous system, your autonomic nervous system is divided into the sympathetic, parasympathetic, and the enteric nervous system. The autonomic nervous system is responsible for what's called our automatic actions, and it is the brain's main system for controlling the body's organs. The sympathetic nervous system and the parasympathetic nervous system act as opposites to one another. The chart on page 39 does a really good job of highlighting that. Basically, your sympathetic nervous system prepares your body for action, which is your fight or flight response. It increases your heart rate and blood pressure and inhibits digestion. It also releases the neurotransmitter norepinephrine, which is produced and released by the sympathetic postganglion neurons to accelerate organ activity, or it's also known as noradrenaline. The sympathetic nervous system consists of sympathetic chains and the nerve fibers that flow through them. These sympathetic chains are chains of ganglia that run along the spinal column. So the parasympathetic nervous system, on the other hand, helps the body to relax, recuperate, and prepare for future action. This is known as the rest and digest period. The parasympathetic nervous system decreases heart rate and blood pressure and activates digestive processes. It also releases the neurotransmitter acetylcholine, which is produced and released by parasympathetic postganglion nerves in response to motor neurons and neurons throughout the brain. The parasympathetic nervous system originates in the brainstem and sacrospinal cord, arising both and above the sympathetic nerves. And then the balance between these two systems determines the state of the internal organs at any given moment. So basically, the tension that ensures in this organism helps it to be prepared for any certain circumstance. The enteric nervous system, not talked about in so much detail, is mainly just responsible for digestion. So it is a mesh-like system of neurons that governs the functioning of the gut, and it controls sensory motor neurons in the CNS, and it maintains fluid and nutrient balances in the body. The spinal cord then sends sensory information from the body to the brain, and in turn conveys the brain's motor commands to the body. The spinal cord contains circuits for local processing and controlled simple behavior and reflexes. Although your brain only weighs about 3 pounds, which is only 2% of the average body weight, it is very intricate. It is actually divided into the cerebral hemisphere, which is basically just the right and left halves of the forebrain. It also consists of the cerebral cortex, which is the outer covering of the cerebral hemispheres that consists largely of neuronal cell bodies, dendrites, and axons. The folding of the cerebral cortex is the reason why the cerebral hemispheres appear to be lumpy. However, the folding of this tissue greatly increases the amount of cortex that we can fit into the skull, as about 2/3 of the cerebral surface is hidden in the folds. The folding pattern, however, it's important to note, is not random. The gyri, which is the ridges or tissues on the cerebral cortex, and the sulci, which is the furrow of a convoluted brain surface, are grouped together in lobes. This therefore forms the four major cortical regions. Each region is named after the bones of the skull that overlie them. This image is found on the top of page 41, which is definitely very important to look into. However, the four major cortical regions are the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. The Sylvian fissure is what divides the temporal lobe from the other regions of the hemisphere, while the central sulcus divides the frontal lobe from the parietal lobe. The physical boundaries of the occipital lobe and the rest of the hemispheric regions are unclear. However, the function of the lobes are quite distinguishable. Your cortex is your main site of complex cognition. So depending on the regions affected, cortical damage can cause severe movement, speech, memory, personality, and visual impairments and changes. Box 2.2 on page 42 is very important to read at your leisure because it does explain the directions for when we're talking about the body and brain. It talks about many key words that are important to know. One main one that I want to distinguish really quick is the difference between an afferent axon and an efferent axon. Remember that efferent axons exit the region that we have in question, while afferent axons arrive in the region that we have in question. On the bottom here, it talks about just different kind of planes. The horizontal plane is responsible for the upper and lower parts of the brain. The sagittal plane bisects the body into right and left halves, and the coronal plane divides the body into front and back. So moving along to the last page, the postcentral gyrus is the strip of parietal cortex behind the central sulcus that receives so much sensory information from the entire body. So the postcentral gyrus is responsible for mediating the sense of touch. It contains a sensory map to the opposite side of the body, meaning that when you feel things with your left hand, it's registered in your right hemisphere and vice versa. The precentral gyrus is a strip of frontal cortex in front of the central sulcus that is crucial for motor control. William Penfield's simulation mapping of the brain showed that the neurons in the precentral gyrus are organized into a map of muscles that corresponds to the opposite side of the body. So it mirrors the postcentral gyrus in the fact that the left hemisphere controls the right side of the body and vice versa. Your corpus callosum, or your main band of axons that connects the two cerebral hemispheres, are C-shaped bundles of actions that enables communication between the two hemispheres, which in turn allows the brain to function as a single unit. So communication flows seamlessly in people with undamaged brains. However, when your corpus callosum is severed, communication between the two hemispheres and the four lobes may become damaged or entirely cut off. When we cut the human brain in half, there are two distinct colors that emerge. The gray matter, or the darker colored exterior, primarily receives and processes information, and the cell bodies are devoid of myelin, which gives them their color. The white matter, on the other hand, is found beneath the outer surface, and it's responsible for transmitting information to other local brain areas. And this consists of mostly axonal fiber tracts that have white myelin sheaths. So I'm sorry this is so long. It is just a lot of information, so I hope you guys took a lot out of it. Thank you.